Hi there and welcome to this IB biology revision video. Mr. Phillips, and we're on video 9, which is looking at cell specialisation, which will look at specialised cells, but also will look at stem cells. And remember, this video is to support you. It is not a full alternative for revision. So as done in previous videos, videos 7 and 8, we're going to be looking at organelles. Obviously you can't look at cells without looking at their particular organelles. And just to remind you, that is what an organelle is, a part of the cell that has a particular specialised function. And there are some parts of cells which we don't class as organelles. So the cell wall, because it's outside of the membrane, the cytoskeleton, because it's spread throughout the cell, it's not a discrete enough region, and the cytoplasm for the same region. And obviously prokaryotic cells have organelles that are not membrane bounded, and they have less functions as a result, but may be more rapid in completion of those roles. So what we're going to look at is stem cells. So stem cells are cells that have an ability to become different kinds of cells. And obviously what happens is a cell becomes more specialised as it develops its ideal structure for a particular role. But it's important to know that all cells have the full 46 chromosomes in the human body, and the embryo is made through mitosis so that these cells can develop anyway. The early embryonic cells are unspecialised, so it's only when you get to this point called a blastocyst that they start to become specialised. And what happens is the enzyme starts to express, the cell starts expressing particular genes to make specific enzymes it needs to become specialised, and then the cell is becoming differentiated. So these genes are switched on or off, which means they are expressed if they're switched on, or not expressed, allowing coding of proteins for specialised cells. And the position of the cell in the embryo is what determines what it becomes due to different chemical gradients present in the embryo. And these are what we call regulators of gene expression. So if there's more of this chemical in a certain place, it might trigger a certain type of cell to develop than if there was less of a chemical in a particular place. Now there's something called the adult stem cell niche, which is where the stem cells are effectively found. And stem cells effectively are cells that can repeatedly divide theoretically with no limits. So skin cells are a good example because they're constantly replacing each other. Testes in males constantly produce sperm cells, so that would be another example. And they're also found in tissues that often need to be regenerated during your lifetime. So as I said, skin cells, liver cells, if your liver is very receptive, it prevents itself getting damaged. Bone marrow cells constantly need regenerating. So the precise location where this happens is called the adult stem cell niche, and it basically is a microenvironment for the stem cells so they can stay there and remain inactive and undifferentiated until needed, because you don't want those stem cells to start turning to other types of cells until necessarily needed. And obviously those stem cells then can become lots of different types of cells. And what will happen if we look at this sort of diagram here is they will basically, until they're needed, they will be just constantly self-renew themselves. So they will stay inactive as long as needed until needed. So a good example is striated muscle cells. They will remain completely inactive until injury, where then they need to replace the muscle cells. Then changes to the niche, so the damage to the niche, which will cause then different chemicals to appear, will cause cells to proliferate and then differentiate, so they become lots of them, and then they differentiate into those muscle cells. Now this use is really important, because what this means is we can store stem cells, we can let them just self-renew and self-replenish until we change their environment, and then they will differentiate to become different types of cells. So in a medical use, this is really useful because we can stimulate them outside of the body by adding different chemicals to make human tissues. And in a non-medical use, we can actually do it potentially for food. So we can use cow striated muscle stem cells in a lab to make edible beef. Now we know that's obviously extremely expensive at the minute, but it's something that in the future may become a useful source of protein. So then there are a couple of different types of cells that you need to be aware of. So there is totipotent cells, which are stem cells that can differentiate into any type of cell. And the typical place you'd say this is from is in the embryo. Now obviously these are very, very useful because we can use them to replace lots of organs, but you have the ethical issues of using an embryo. It can't give consent, and if you use it, you're going to basically harvest the embryo and then kill the embryo. So some people would say that would be murder, and it's therefore unethical. Pluripotent cells are still capable of differentiating into a range of cell types, and typical example this will be in your later embryo. And then multipotent cells are the ones probably with the least ethical issues because you could get them from yourself. Adult stem cells basically can become several types of cells, such as the hemopoietic cells in bone marrow, which can generate the different types of blood cell that you have. So if you look at that, we've got totipotent can generate the most cells, which is most useful, but probably the most ethical question, and then the least ethical question doesn't generate as many cells, as a result may be less useful. So there are two main sources: human embryos, which can differentiate into most cell types, but we have those ethical issues of a lack of consent and killing the embryo, and adult bone cells, which can differentiate into some cell types, but it needs to get it from the bone marrow, which is a painful procedure, but there aren't the ethical issues because you can obviously give permission to do so. So you might have to talk about this for use in something called therapeutic cloning. And in theory, if I needed a new liver, I could, well I can't because it's illegal, but in theory, scientifically, I could clone myself into an embryo and then use those embryos to get stem cells for myself. But obviously this is something that has a lot of ethical issues, and we are obviously trying to push the use of adult stem cells, you can see here, to create different types of cells and make this less ethical. So that's your real key. So if we start looking then at cell differentiation and cell specialization, though, what actually happens and how do those cells become specialized? So the first thing here is you need to understand what is happening when differentiation occurs. So cells become specialized to carry out particular functions in multicellular organisms, which makes them more efficient. So you need specific cells doing specific jobs, otherwise you wouldn't survive as a multicellular organism of billions of cells. Each cell has about 4,000 housekeeping genes, which code for proteins that do all the reactions the cell needs to survive, such as respiration. The rest of the genes are only expressed when they're needed to carry out a particular type of role. So for example, the pancreas cells are the only cells that express the insulin gene to create insulin. It wouldn't be created, that gene does exist in your liver cells, but it isn't created in your liver cells. It's created only in the pancreatic beta cells. Gene expression occurs early in human embryos and development of cells as they differentiate and they make specific proteins to become specific for their role. So very early on, this differentiation process occurs to create your brain cells, your kidney cells, your liver cells. Now, there are some atypical eukaryotic cells that you need to know about, and you need to know the function and how they are particularly adapted, why they're atypical, why are they different from typical eukaryotic cells. So the first type is red blood cells, and you'll know this from earlier on in school that red blood cells have no nucleus. And if you look here at the development of red blood cells, they start off obviously as multipotent stem cells, we just said, in the bone marrow, and at first they do produce, they do have a nucleus, and then what happens is the nucleus is moved to the edge of the cell and it is then pinched off in a vesicle, and then a phagocyte destroys that. White blood cell destroys it, so then the final red blood cell has no nucleus, which allows it to carry more oxygen and have more hemoglobin in it. So the red blood cell initially does have a nucleus, but that is ejected out and destroyed by a phagocyte. And this is good for red blood cells, it makes them more flexible, but they cannot replicate, so have to be replaced every 100 to 120 days. In plant cells, we've got the phloem sieve tube element, and basically these are adapted by having to allow sucrose to move up and down the plant. They have to have holes in their cell wall. So the cell wall at the end there, you can see, has holes in it, allowing sucrose to move through it. This is called a sieve plate, and the holes are obviously sieve plate pores. Now, because of this, and because of the fact they want sugars to be able to move freely unprevented through the tubes, they have a massively reduced cytoplasm. So that tiny sort of yellowy brown ring, like line there, is all they have as a cytoplasm. The green bit is completely empty, and what that is allowing is obviously lots of sugars to move up and down the plant. But the only way they can therefore be alive, and they do need to be alive for this, is they have to have a companion cell next to them which does all the processes. So we'd say a companion cell has a thick cytoplasm or dense cytoplasm with extra mitochondria, with extra chloroplasts, with extra whatever it needs, ribosomes, probably not chloroplasts, with ribosomes, to keep that cell alive. And in actual fact, the reason they're called sieve tube elements and not sieve tube cells is because they lack all the typical contents and rely on the companion cells to be alive. Skeletal muscles, which are your muscles that connect to bones effectively and allow movement. So like if you move your arm, the tricep and the bicep, they are large multinucleic structures. And basically what happens is they start off as separate cells. So imagine we've got a load of separate cells here, okay, and those cells are obviously separate. They will all have a nucleus to start off with, but then what happens is the cell membranes between them break down and they fuse into one type of cell. So the membrane will fuse into one type of cell as the cell as it matures, and this means they have long fibers, and they need to be long because they need to be able to contract and then relax and get longer again for muscle to do its job. The last one is a hyphae, which is part of a fungus, fungal hyphae. And normally what happens is we have, or not normally, a lot of the time what happens is there is what we call a septate hyphae. You can see these are individual cells and they have something called a septum, which is a wall between them. Okay, but some fungus develop to be aseptate and aseptate, I should say, and that you'll notice the wall has gone, and this means they have large nucleic structures called acenocyte as a result, so large multinucleic structures. And again, it will mean that they can grow without the impediment of flow between one cell to another. So this is all really important, and it has allowed, this specialization of cells has allowed multicellular organisms to be created. So all animals, plants, many fungi and eukaryotic bacteria, algae I should say, are multicellular. Most cells can no longer live independent. So why is this so important? Well, it means you have a longer lifespan, as the death of a cell does not prevent survival. If you think about it now, sat where you are in your seat is not the same cells that were sat there when you were born as a baby, and by the time you're 30, those cells will have been completely replaced again. So lots of your cells are constantly dying, you don't even know about it, and that allows longer survival rates. Bacteria do not live long because they don't have multicellular. Generally, it means you're larger, so you can exploit niches that other unicellular organisms can't, and increased complexity because you have differentiated cells doing different jobs and then working together to be part of the organism. But whilst this is important to remember, and it is important because it means that we're more advanced in many respects, be aware that most of the Earth's biomass is unicellular. So all bacteria on the planet would weigh more than all the multicellular organisms on the planet. So they have survival advantages, such as being able to mutate regularly, being able to rapidly reproduce, as opposed to what multicellular organisms can do. So we talked about what an organelle is earlier. You also need to be able to talk about then how some cells are specialized. So we need to know how some cells are specialised, particularly due to their size. So if you look at these different cells, they all have different sizes. So the sperm cell is obviously extremely, 50 micrometers long, but it's narrow, and therefore it also has a small volume. This is obviously used because it needs to swim to the egg, so it reduces resistance or friction within the female liquids, and therefore it can get to the egg easier. The egg has a large diameter, it's the largest human cell, and obviously it's got food reserves in it to help the cell grow and survive before it implants into the uterus lining and develops a placenta. The red blood cell is quite a narrow diameter, only 8 micrometers in diameter, which is extremely thin, and 1 micrometer thick in the middle, and this obviously means that it can have a large surface area. The small size means it can fit very packed and closely through the capillaries, allowing easier unloading and loading of oxygen. White blood cells are bigger cells because they get bigger when they're active, and that is because they have lots of rough endoplasmic reticulum and Golgi apparatus making proteins such as antibodies, so they need lots of them, dense cytoplasm full of those things. But also phagocytes will be doing phagocytosis, so they have to engulf smaller bacteria as well, and then obviously break them down by hydrolysis. Cerebellar granule cells, which are basically neurons, are very, very small, and the reason they're very small is there are 50 billion of them in your brain, so therefore there needs to be lots of space for them. They need to be small to allow it to happen. Motor neurons are very long, so the motor neuron part of the reflex arc, so it's the neuron that sends a message from your spinal cord down to the effector, so they have to go maybe all the way down the length of your arm to pass that message along. And muscle fibers, striated muscle fibers, change lengths so they can exert a force through contraction. So surface area to volume ratio can have constraints on cells, and therefore cells are often adapted to increase the surface area to volume ratio. So cellular reactions may occur in the cytoplasm, and some of those reactions is called metabolism. Metabolic rate is directly proportional to the volume of the cell. So the bigger the cell, the more metabolic rate there needs to be. But it is limited by surface area because you need to obviously get chemicals in and out for metabolism to happen. So for example, you need oxygen to come in for a cell to do respiration. And if you've got a small surface area, then not enough substances can move in and out, and the heat produced might not be able to escape, and also you might have a build-up of toxins or byproducts that you don't want. So cells such as the root hair cell, which you should be familiar with, has a large surface area to get more materials in, and obviously that's for absorption, particularly of minerals from the soil, and digestion cells have large surface areas. You need to be able to calculate the surface area to volume ratio. And a lot of students get confused with this because they seem to try and do the opposite way around. But if you think of this as literally being surface area divided by volume, that is the best way to do it. So if we have a look at, we'll look at this cube here, the medium size cube. So if the medium size cube has got each side is 5 centimeters in length, the surface area of that cube is going to be 5 times 5 to get that, which is 25, but then it's the whole surface area. So don't forget there are 6 sides to each cube, so that gives 150 centimeters squared surface area. And if you remember how to calculate volume, volume is length times width times height, so 5 times 5 times 5, which gives us 125. So then to work out that number there, I would do 150, the surface area, divided by 125, which gives me 1.2. Now just be aware, a lot of people forget to multiply it by 6 to calculate the surface area. And this one, you've got a large cube of 10 centimeters, so you'd be doing 10 times 10 times 6, so 600 to work out the surface area, and then 10 times 10 times 10 to give us 1,000 to work out the volume. So then 600 divided by 1,000 equals 0.6. Now you may be given it like this in a cube, and you might be able to do all of these different ones. So if you struggle with this, pause it here and make sure you're okay to do it. But in real life, a cell isn't going to be a cube. So what we'll often use is a sphere, and the biological assumption is the cell must have a universal shape. Obviously, this gives us an approximation, but it isn't actually 100% correct. There will be realistically, the cells are not this perfect sphere shape. And here you can work out surface area by using area equals 4 pi r squared and volume equals 4 over 3 pi r cubed. So again here, if you've got 10 micrometers in diameter, remember we're working out radius, so it's going to be 5 because radius is half of the diameter. So what you can do here is work out the surface area of the cell first of all. So you're doing 4 times pi times r squared, 5 times 5, which equals 314. You then work out the volume of the cell, which equals 3/4 times pi times 5 cubed, 5 times 5 times 5, which equals 523. And then just like before, I'm going to do 314 divided by 523. Okay, now they've presented it here as 0.6 to 1. Okay, you could just put that as the decimal number, which is about 0.6. But if it depends how they ask you to do this, and if they give you other examples within in a table, for example, if they want it to 1, it would just be 0.6 to 1. So you need to know how some cells have adapted to increase their surface area volume ratio. So red blood cells, you should know, are extremely small and they have a concave shape, a biconcave shape, should really say, to increase their surface area to volume ratio. You will study the kidney tubules later on. Okay, so the kidney proximal convoluted tubule is in the kidney, and basically what is happening is the fluid from your blood is moving down these tubes, and as they move down these tubes, important materials such as water are reabsorbed back into the bloodstream, and sugars, so glucose is reabsorbed back into the bloodstream. So they have a large surface area to allow them to increase their reabsorption back into the bloodstream, so you don't lose lots of important chemicals and too much water through your urine. They have lined with microvilli to increase the surface area, and they have proteins in there to pump ions into this area here and this area here to allow water to move out via osmosis. So you also need to be able to research and talk about the following cell types: so alveoli, okay, cardiac muscle cells, and egg and sperm cells. So if we have a look at these, first of all, the alveoli, obviously, as you know, is part of the lungs, so it's important in gas exchange, lined with capillaries, and therefore they need to be extremely thin to allow oxygen to diffuse out of the alveolar space here into the capillary and into the red blood cells, and carbon dioxide to come back. And basically the alveoli have different types of cells called pneumocytes. So the type 1 pneumocytes are what you would have talked about previously in school, very, very thin cells that are adapted for diffusion of oxygen and carbon dioxide. There's very few mitochondria, very few organelles, very few tiny cytoplasm, but very, very thin cells as a result. So it allows this movement of oxygen across, let's say, into that cell. And here's a good example here. You can see how thin this is compared to the type 2, which we're going to look at. So it would move through here and into the lining of into the red capillaries. However, they need to be kept alive again. So type 2 pneumocytes are more numerous but only contain 10% of the area. They have a dense cytoplasm and they produce vesicles called lamellibodies, which are released via exocytosis. So they have a key role in the maintaining of the health and the living part of the alveoli. So when we look at these, we need to understand what is going on. So I just said about phospholipids being released. We'll talk about that now. So the alveoli is lined by moisture, allowing oxygen and carbon dioxide to diffuse in and out of the capillaries. The phospholipids are released from the lamellibodies, line the outer surface on moisture and a single layer, and proteins are dispersed between them. So you see here you've got this layer, these phospholipids are released and they actually lie along the outside of the alveoli. And the tails stick outwards. And the importance of this is why these pneumocytes type 2 are so important, is they release this phospholipid layer, and this phospholipid layer, the tails sticking outwards, prevent water from forming along here, because the hydrophobic tails are on the outside. It prevents water molecules adhering, so it stops the collection of water in this area here where it could cause the alveoli to collapse. So you have two types of cells. Type 1 is the very thin type, okay, the pneumocyte type 1, where the oxygen and carbon dioxide can diffuse over without any problems. And then you've got type 2. Their key role is to release these phospholipids, and they have a hydrophobic tail which then prevents water adhesion, prevents the water from cohesing up and causing the cells to collapse, the alveoli to collapse. Cardiac muscles are the only muscles that can contract, and you do need to research these and look at how they are different from normal skeletal muscles. I'm not going to do that in this video because it will mean the video goes over 30 minutes. And then we have egg and sperm cells. Both of these are haploid cells, which means they have half number of chromosomes in, so 23 chromosomes in humans as opposed to 46. So they can fuse to make a diploid cell of 46, which is the zygote, the first egg. And the egg cells are adapted to only be fertilized by one sperm. So often when you teach this lower in school, you always get the question, what happens if two sperm get in? Two sperm cannot get in because of their adaptations. So they have got different layers. So the first layer is this zona pellucida here, which is this circular, my pointer's going around now. And this is a layer of glycoproteins containing ZP3 receptors, which is what the sperm attach to. And as soon as the sperm attach, they can then be later chemically altered. They have binding proteins on them, which again the sperm can attach to, and cortical granules, which are vesicles of enzymes near the plasma membrane of the sperm, released into the zona pellucida, making it impenetrable as soon as the sperm enters. So basically what happens is when the sperm enter here, so imagine your sperm actually getting through, as soon as that sperm gets through, the chemicals are changed here, preventing this bit from being penetrable after that point. And it is the cortical granules, which are this bit here, okay, which enter the membrane, vesicles do, as soon as the sperm enters, making it impenetrable. Now obviously, basically egg cells also have some basic adaptations. Yolk, which stores lipids, so fats and other food supplies, energy resources to help the early egg grow before it implants into the ovary. And that's why I think animals that just lay eggs need bigger eggs even more, because they don't have that food supply, so it needs to last them until the baby hatches out, such as birds, for example. The mitochondria, they've got lots of mitochondria to provide ATP, which divides to make all ATP in future cells. So that one mitochondria becomes all the mitochondria that you will later see, and centrioles used in mitosis. And obviously they have to be in there, the centrioles, because they need to make spindles for mitosis. And at this point, this cell has to divide. And how is it going to divide? Via mitosis. So they have to be ready in the egg cell, ready to begin. The sperm cell is much more adapted just to provide that genetic information. So the genetic information in the sperm cell is all about getting to the egg. So it is adapted to compete and hopefully beat the other sperm cells to get to the egg, which means only the strongest sperm cells survive. It is a tail made out of what we call 9+2 microtubule arrangement of a flagella basically, which allows it to have forward motion through beating. It has a mid-piece in it with mitochondria in it, providing ATP to allow it to respire and to get to the egg. It has a head which is streamlined and tightly packed, having a small cytoplasm to allow it to burrow into the egg, and it has receptors on the head of the sperm cell, which is receptive to those ZP3 glycoproteins that we mentioned a few minutes ago in the egg. It has an acrosome which releases enzymes that help hydrolyze the polysaccharides in the zona pellucida to allow it to reach into the plasma membrane, and it has binding proteins that allow it to attach to that plasma membrane in the first place to allow then the egg cell to remember. So key adaptations that you need to know, and don't forget to research your muscle cells yourself, because I have now run out of time.